a window into gene expression introducing Lampbrush chromosomes. Lampbrush chromosomes are fascinating structures found within the cells of most animals, with the notable exception of mammals. These chromosomes are unique to a specific stage of cell division called meiosis, specifically the diplotin stage. During this time, the chromosomes are preparing for the critical process of reducing their genetic material by half before sexual reproduction. It is in this preparatory phase that Lampbrush chromosomes become visible, showcasing their distinctive morphology. Their presence signifies a period of intense activity within the cell, particularly in the synthesis of RNA, a crucial molecule for gene expression. Lampbrush chromosomes provide scientists with a unique opportunity to observe and study the intricacies of gene expression. Unlike typical condensed chromosomes, Lampbrush chromosomes are highly extended, resembling a tangled mass of threads adorned with numerous loops. These loops, emanating from the main axis of the chromosome, are not random structures, but rather represent regions of active gene transcription. In essence, they serve as visual indicators of where genes are being actively copied from DNA into RNA, the molecular messenger that carries genetic information from the nucleus to the cytoplasm for protein synthesis. The study of Lampbrush chromosomes has been instrumental in our understanding of how genes are regulated during development. Their unique structure provides a visual representation of the transcriptional landscape of the cell, highlighting the spatial organization of genes and their activity levels. By analyzing the size, shape, and distribution of loops along the chromosome, researchers can gain insights into the dynamics of gene expression. Moreover, Lampbrush chromosomes offer a valuable system for investigating the role of non-coding RNA molecules in gene regulation. These RNA molecules, once considered mere byproducts of transcription, are now recognized as key players in various cellular processes, including chromatin organization and gene silencing. The anatomy of a Lampbrush chromosome: loops and transcription factories. Lampbrush chromosomes, named for their resemblance to the brushes used to clean kerosene lamps, are marvels of cellular architecture. Their most striking feature is the presence of numerous lateral loops that extend outward from the main axis of each chromosome. These loops, far from being mere structural appendages, are the sites of intense RNA synthesis, highlighting their crucial role in gene expression. Each loop represents a single DNA strand that has uncoiled and extended outward from the densely packed chromatin fiber. This uncoiling allows the cellular machinery responsible for transcription, including enzymes like RNA polymerase, to access the DNA sequence and initiate the process of RNA synthesis. The loops of Lampbrush chromosomes are not uniform in size or shape. Their dimensions can vary significantly, with some loops extending hundreds of times the diameter of the main chromosome axis. This variation in loop size reflects the transcriptional activity of the underlying DNA sequence. Larger loops are generally associated with genes that are being transcribed at a higher rate, indicating a greater demand for the corresponding RNA molecule. Conversely, smaller loops or the absence of loops suggests lower or negligible transcriptional activity. This direct correlation between loop size and gene expression makes Lampbrush chromosomes an invaluable tool for studying the dynamics of gene regulation during development. The loop structure of Lampbrush chromosomes is not static. These dynamic structures constantly undergo changes in size and shape, reflecting the fluctuating demands of gene expression during oocyte development. As the oocyte matures, certain genes may be upregulated or downregulated, leading to corresponding changes in loop morphology. This dynamic nature of Lampbrush chromosome loops highlights the intricate interplay between chromatin structure and gene regulation. The organization of DNA into loops and their subsequent modifications play a crucial role in determining which genes are accessible for transcription and at what levels. A legacy in microscopy unveiling the world of Lampbrush chromosomes. The discovery of Lampbrush chromosomes dates back to the late 19th century, marking a significant milestone in the history of cell biology. In 1882, Walther Flemming, a German cytologist, first observed these peculiar structures within the nuclei of salamander oocytes. Intrigued by their unique appearance, resembling the brushes used to clean kerosene lamps common during that era, Flemming coined the term Lampbrush chromosomes. This discovery marked the beginning of our fascination with these intricate structures and their role in gene expression. Early observations of Lampbrush chromosomes were limited by the rudimentary microscopes available at the time. However, these initial studies laid the foundation for our understanding of their basic morphology and hinted at their potential significance in cellular processes. The advent of more powerful imaging techniques such as electron microscopy in the mid-20th century revolutionized our view of Lampbrush chromosomes. Electron micrographs revealed the intricate details of loop structure, showcasing the DNA axis adorned with nascent RNA transcripts and associated proteins. These high-resolution images provided compelling evidence for the role of Lampbrush chromosome loops as sites of active RNA synthesis. The development of fluorescence microscopy further enhanced our ability to study Lampbrush chromosomes. By labeling specific DNA sequences or RNA molecules with fluorescent probes, researchers could visualize the spatial organization of genes and their transcripts within the context of the chromosome. This technique enabled the study of chromosome dynamics, such as loop formation and movement, providing insights into the regulation of gene expression during development. Beyond the microscope, modern tools for studying Lampbrush chromosomes. While microscopy remains an essential tool for studying Lampbrush chromosomes, recent advances in molecular biology and genomics have provided unprecedented insights into their structure and function. Techniques like chromosome conformation capture, 3C, and its derivatives, 4C, 5C, high C, have revolutionized our understanding of the three-dimensional organization of chromosomes within the nucleus. These methods allow researchers to identify regions of the genome that are physically close to each other, even if they are located far apart on the linear DNA molecule. By applying these techniques to Lampbrush chromosomes, scientists can map the spatial relationships between different loops and their associated genes, providing insights into the regulation of gene expression. Another breakthrough in Lampbrush chromosome research has been the application of RNA interference, RNAi technology. RNAi is a powerful tool for silencing specific genes by targeting and degrading their corresponding messenger RNA molecules. By applying RNAi to Lampbrush chromosomes, researchers can investigate the functional consequences of silencing specific genes during oocyte development. This approach has been particularly fruitful in studies using the fission yeast, a model organism that shares some similarities with higher eukaryotes in terms of chromatin organization and gene regulation. Recent advances in imaging technology continue to push the boundaries of Lampbrush chromosome research. Super-resolution microscopy techniques, such as structured illumination microscopy, SIM, and photoactivated localization microscopy, PALM, are capable of breaking the diffraction limit of light, allowing for the visualization of cellular structures with unprecedented detail. These techniques have enabled researchers to study the dynamics of Lampbrush chromosome loops with greater precision, revealing the intricate interplay between DNA, RNA, and proteins during transcription. As imaging and sequencing technologies continue to improve, we can anticipate even more exciting discoveries that will further illuminate the secrets of these remarkable structures.